pretty soon. So no, just keep going. Reading audio may not be, well, something tells me. Right, okay, yeah. Okay, speaker's fine, speaker's fine, speaker's fine. Okay, great. All right, so now back to the ranch. There we are. I'm well aware of that. There, boy, that takes a lot longer than it should take. So skeletal muscles, when they are born, you have these cells called myoblasts. And the myoblasts undergo a massive round of fusion. And so you get this large multicellular cell that's called a myotube. And that myotube begins to differentiate. And the cytoplasm gets filled with contractile elements that are organized in a very regular fashion. And the nuclei get pushed to the periphery, and then the cell extends in either direction. And it finds places to attach, and there are usually areas marked on the skeleton or skin or wherever it's going to attach. That tell it to attach, it then attaches, and tension is actually required for the rest of its development. But consequently, you have a long cell that has these arrays of contractile elements, and those array of contractile elements repeat in a very regular fashion. And those repeating units, it'll be on the slide here in a bit, but they're called sarcomeres. And the sarcomeres consist of actin and myosin, and you have the heavy bands and the light bands, and actin makes up the light bands. And myosin composes the heavy bands. Now, these aren't the only proteins in those light and heavy bands, but they're the main proteins in the light and heavy bands. Myosin.
That's just
Whole heck of a lot of sense there.
I looked up and I was like,
Heavy or sometimes thick. Thick bands. Muscles are connected to usually the skeletal muscle, but some, for example, the muscles that are involved with facial expression, they're attached to the skin. So whatever they're attached via a tendon. And as we'll see, the muscle has this very sophisticated covering on it. That covering essentially is going to go to the end of the muscle, and then it's going to join and it's going to form the tendon. It's very common to talk about tendons as just being big, long cables of collagen. They're actually a lot more complicated than that. There are cells in there as well, and those cells are actually quite important in the maintenance and the repair of the collagen, as it turns out. So the tendon is going to cause the movement of the joint. When the muscle attaches, it'll move the bone this way, that way, whatever. And you're going to have one of those attachment sites that doesn't move very much relative to the other. And so the side that the attachment site that moves more, the other one, that's the insertion. The other type that's relatively stationary is the organ. And the skeletal system is moved essentially by the contractions of those skeletal muscles. Now, this is again is old hat to those of you in HES classes. These are all words that you heard and probably use a lot in your everyday vocabulary when you're talking about your field. So extensors will increase the angle at a joint. Flexors are going to decrease the angle at the joint. So extensors, flexors, when you think about the hand. Abductors move things toward the midline. With the hand, of course, it tests with the middle finger. If you move it toward the middle finger, that's an adductor of the hand. Abductors move things away from the midline, and the mnemonic there is you're adding to the diameter by moving it toward the midline. That's the mnemonic, and then ab is the opposite of add. Levators will move insertions up, depressors will move it down. Rotators will allow pronation, supination along an axis. And then sphincters very often are smooth muscle, but sometimes a lot of your sphincters, particularly ones that open to the outside, are dual. And you have one sphincter that is autonomically controlled, the other that's under conscious control. So some sphincters are under conscious control. They are voluntary muscle, skeletal muscle. The other sphincter is not. So you'll have an internal and external sphincter, one under conscious control, the other that's not. Well, a lot of your other sphincters are not under conscious control. They're composed of smooth muscle, not skeletal muscle. So when it comes to the muscle, the individual muscle cells themselves contain these repeating sarcomeric arrays. And those sarcomeric arrays, one of those essentially, one bundle of those sarcomeric arrays, they constitute the so-called myofibrils. And as you can see, the individual muscle cell itself has several of these bundles, these myofibrils in them. Because the cytoplasm of the muscle cell is pretty specialized in its organization and its composition, it gets referred to as the sarcoplasm. That just simply means the cytoplasm of a muscle cell. The endoplasmic reticulum of a muscle cell is largely modified into a calcium storage organelle. It's also found at junctions. So the membrane of the muscle cell is also very specialized because it has these indentations in it that form these tubules that run across the membrane, and they're called transverse tubules or T-tubules. And the sarcoplasmic reticulum is always organized around those T-tubules. Because of its unusual function and organization within the muscle cell, it gets called the sarcoplasmic reticulum. It sounds like anatomists just finding new ways to torture students with new terminology. But it's given that terminology because it's unusual. So I think that marks it off, and by marking it off, you can see it does something special vis-a-vis muscle cells. So the outside of the muscle consists of this very tough covering fascia. If you're Bethel Rick, you say fascia, but I've always heard it say fascia. So the fascia, as you see, is pretty contiguous with the tendon, and it does cover the tendon. Fascia is not just fibers, there are cells in there too. And the cells often require tension for their health and welfare, for their function. Same thing with the cells in the tendon, the so-called tenocytes. Tenocytes, as it turns out, they respond to tension. And that's why when you injure a muscle and you get it worked on, the surgeon will immobilize the muscle. But I'm convinced half the recovery of the muscle is not just the healing, it's actually getting that tendon that's degenerated while it was immobilized back to it. That's why, and Mitch and I have talked about this at length. I think a lot of surgeons now are no longer slinging shoulders and things like that. You're just supposed to take care of it, not move it very much, but you're still supposed to move it a little bit to keep that tension, because without that tension, those tenocytes start to become quiescent and they don't maintain the tendon anymore, and the tendon gets weak and what have you. That's kind of a completely off subject sort of topic. Yeah, Bracken.
Is the fascia under voluntary control, or is it just a covering?
Fascia is pretty much a covering. It responds to the same tension as the muscle. So beneath the fascia, you have a series of coverings, as you can see, of the different muscle cells. So each of those muscle cells, with the muscle cells that contain the myofibrils, and the repeated organization of those sarcomeres, the zebra stripes, those are called striations, hence the term striated muscle, striped muscle. So if you look essentially at those muscle cells all organized together in that unit is covered by a covering called the paramecium, and then the paramecium is going to also dive inside and surround each of those muscle cells. Where it surrounds each of those muscle cells, that's called the endomecium. And then the covering around the whole muscle, that's the epimecium. This is all composed of collagen and extracellular matrix and stuff like that. So if we look essentially at this cell, you can see the nucleus here. So we've taken one of these, we've blown it up. The nuclei are peripheral. That's really common in skeletal muscle. The nuclei are not in the middle of the cell like they are in a cardiac muscle cell. They're always peripheral. They get squeezed to the edge and they're always kind of bean-shaped. And you have this constant repetition of those sarcomeres. Now, those sarcomeres are going to contract. And as those sarcomeres shorten, the whole muscle is going to shorten, and that is the whole nature of contraction. We are going to consider muscle contraction by looking at just one sarcomere, but you must remember, all the other sarcomeres are doing the same thing. That's what's going to cause the whole muscle to contract. So if we look at it even a tad bit closer, you can see the light and heavy filaments here made of actin and myosin. Those are those myofilaments. And in your cross section here, the little dots, those are sometimes called myofibrils. And those are organized into these nice little contractile elements that are very regular looking. And that's a feature of skeletal muscle when you look at it, particularly in the electron microscope. It's really, really regular looking, really, really highly organized. If it's not, then something's wrong. Because the muscle, if it's not highly organized, it's not going to work properly. So now let's take a look at these arrays using low power transition electron microscopy. And we can see this regularity even in more living color. So we have these areas here, light areas, and then we have the heavy bands here. So from the end of this heavy band to that, that's called the A band, And each sarcomere then is bordered by this dark staining Z band. And my really dumb mnemonic is that the sarcomere goes from Z to shining Z. That's how you remember the Z band. The Z band is full of integrins. Integrins are very important for those sarcomeres to be able to attach together. There are muscle-specific integrins in there. Desmin is one of them. There are some others. And they form essentially the Z band where the light bands are going to, the actin filaments are going to be anchored and then stick towards, jut out towards the myosin and the A band there. Now there is a band that goes down the center of this. That is called the M band. The M band. Now the M band, M is a good name because M stands for mirror. And when you think about this, if you cut the sarcomere in half, this side is a mirror image of that side. So the M band is right smack in the middle of the sarcomere, and it's a reflection. The other side is a reflection of the opposite side. That M band consists of a molecule called myodesmin. That's in the M band. And we're going to see the role of the M band planes here in just a bit. So if we take the molecules that compose the elements of the sarcomere and put them in a cartoon, so they're a little easier to see, we also make it more 2D instead of more 3D, you can now start to get an idea of how these are organized in, I think, a pretty unique and abiding way. So notice that the actin microfilaments here are embedded in the Z disc here. There's a protein rod actually that this winds around called nebulin. Nebulin makes sure that this thing is not going anywhere. It's also capped at the end over here by a capping protein. This actin filament is essentially made to last. It's no actin usually in cells is pretty transient. This actin ain't transient. It's made to last. Yeah.
Z disc, is that the same thing as Z band?
Yep. In 3D, it's a disc. In 2D, it's a band. Because the sarcomere is a three-dimensional structure. But when you draw it 2D, you get the idea the sarcomere is flat. The sarcomere is cylindrical. It's 3D.
Does the I band extend from the red portion until it would touch another red portion?
Yeah, so that I band essentially includes all the area where myosin is not. And the A band includes all the area where myosin is. So you take two halves of the I bands and the A band in the sarcomere, essentially. And the M line, the M band is going to go right in between these arrays of myosin there. And you can see it's a mirror. It essentially is a mirror between the two sides. So if we strip it all down to just lines and give those lines little different colors, we have the actin filaments in the purple here and the myosin in the green. We can get a pretty good idea of I bands, A bands, H bands, and what have you. So the H band is that area between the two actin microfilaments on opposite sides of the sarcomere. Now, when this muscle contracts, those two ends are going to move towards each other. And the two Z discs are going to move towards each other. So that's essentially how physically the sarcomere is going to get shorter. And that's going to happen for every other sarcomere attached to it. So we're going to detail it for just one, but this is also happening to all the other sarcomeres as well. And this picture here, which is from your book, I really like this picture because it tries to convey the idea of how 3D this structure is. And then I think that might be what you were getting at with the question. This shows you essentially the 3D structure, and you can see the microfibrils in this picture here And that shows you essentially the 3D structure. So it's harder essentially to detail this if we do it like that. So we're just going to do the 2D picture, and maybe someday someone will design a program where everyone can wear an Oculus, and then we can somehow, you can with your hands, you can do this, and with the sarcomere contract. I think that'd be a great program for someone to write. I'm not going to write it because I don't know anything about computer programming. But that essentially, the 2D that we're doing is meant to represent something that's actually 3D. Before we get to that whole contraction thing, let's say something about the nervous system, because the muscle is not really going to do that unless you have a nerve that tells it to contract. So the nerve that tells it to contract is a motor neuron. A motor neuron. Its cell body is in the spinal cord, and it is going to issue from the ventral side. The axon issues from the ventral side of the spinal cord. Its neurotransmitter is acetylcholine, and its synapse with the muscle is via a very special structure known as either a neuromuscular junction or a motor end plate. Now, neuromuscular junction can be a term really for any nerve muscle junction. So it can be for autonomic nerves that have junctions with smooth muscles. When it comes to motor neurons and skeletal muscles, that junction is very specialized, so it's called a motor end plate. A motor end plate. It's a very, very special structure. Let's take a look at this very special structure. I love this picture. That beautiful SEM picture. Notice how it's like the nerve almost excavates the muscle. It's actually worked. It's like it's burrowed into the muscle almost. And it's almost flush with the surface of the muscle. So this connection between the motor nerve terminus, motor neuron axon terminus, and the muscle is very intimate. Very, 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 very intimate. Okay, story. I was a TA at UC Irvine, and Irvine, after the Vietnam War, Southern California was an area where there were lots of Vietnamese refugees that were resettled. And Garden Grove, California there's an area in it called Little Saigon, because if you want good Vietnamese food, that's a place to go. Best Vietnamese food there, and it's all delicious. Anyway, I was telling them about things that intimately pair with each other. And the Vietnamese girl raises her hand and back. It's about chromosomes intimately pairing. She says, how do chromosomes have sex? I went, oh, intimate. She took the term intimate to mean that kind of intimate. It just goes to show you sometimes how language sometimes can be more of a barrier than we wish it was. But so the association between the two is quite intimate. Now, if we turn this into a cartoon, we can see here that in fact that is true, that the membrane of the muscle cell has indentations to receive the branches of the axon terminus. Furthermore, not only is the membrane indented, but you have further infoldings of the sarcolemma around this motor neuron axon terminus, filling the cytoplasm of this motor neuron axon terminus are, of course, lots and lots of synaptic vesicles that are laden with acetylcholine. So this particular structure, this whole structure here, this whole thing is called the motor end plate. The motor end plate. That's what we mean by the motor end plate. The axon will tend to have lots of collateral branches. It will tend to innervate several different groups of muscle cells. And the muscles that are contracted when a particular motor neuron fires is called a motor unit. A motor unit. So, okay, now go ahead and go ahead, Flex. Okay. That doesn't take much effort. Okay. Now let's okay. Now, was that's still pretty easy, but was it as easy as this? No. Okay. Now do it. You got to more, you got to recruit more motor units. Okay, that's an example of recruiting motor units. Something that requires more power, requires more motor unit, and you're recruiting motor units. That's how you get more power. It's more motor unit recruitment. Weightlifting increases the size of the muscle cells. So it takes fewer motor, you have to recruit fewer motor units to lift the same amount of weight. That's why certain weights now don't seem as heavy, because the actual units themselves consist of more muscle, larger muscle cells that can do more work, essentially. That's the benefits of weight training. Thanks for being a good sport there, Bracken.
Good morning.
All right, your book has this really nice figure that essentially illustrates nicely what a motor unit is. You can see, you can see essentially how the motor termini, they're going to innervate all these fibers here. So all these fibers here, that constitutes a motor unit. And we're going to do a lab using BioPack next week, where we're going to use a dynamometer, and you're going to see essentially how the signal you get from the muscle as you contract it, when you contract harder, that signal now becomes more robust, because you're having more depolarization of muscle cells as you demand more from those muscles. You have to recruit more motor units. And if you take those axon termini and their neuromuscular junctions and blow them up, this is essentially what you're getting essentially when you act these particular muscle fibers. So there is a particular ratio that determines essentially what kind of fine motor control you will have over muscles. Large muscles that give you lots of power usually have very low innervation ratios. Other muscles that are very good for fine movement, but don't give you a lot of power, tend to have very high innervation ratios. So innervation ratios simply have to do with how many motor units get innervated per motor unit. So if you have more more nerves essentially innervating a smaller muscle, less power, more control, better fine control. If you have fewer nerves, but those fewer nerves control a lot more muscle fibers, you get less fine control, but a lot more power. So you can't have one without the other, essentially. You can have one, but not the other. So if you think of the muscles that position the eye, essentially, the innervation ratios in those are like one neuron for every 3 muscles, essentially. Whereas your calf muscle, the gastrocnemius, that has a neuron essentially per 1,000 muscle fibers. So the innervation ratio can vary pretty substantially, So if you consider the muscles, say, in your upper arm and compare them to the muscles that control your fingers, which you're going to have a higher innervation ratio. Your fingers, relative to your deltoid or bicep brachii or brachialis or tricep or something like that. All right, any questions about any or all of that before we go on to microstructure and what happens with microstructure when it contracts? Good, all right. So in the vertebrate, you have three different types of muscles. Skeletal cardiac smooth, where all about the skeletal essentially in this unit here. So the fiber is one fiber in a bundle of myofibrils, and the muscle fiber consists of one muscle cell, and that muscle cell, at least in humans, is about 50 microns in diameter, but it's very long because remember, it's a consequence of several cells fusing. And the myofibrils themselves have these repeating subunits called sarcomeres. Those sarcomeres are 2.3 or so microns long-ish, something like that. So here we have some very high resolution pictures of muscle structure. And you can see this very nice picture here. That's a light microscope picture of the sarcomeres. You blow that up essentially. You get essentially the sarcomere structure here, which has the structure that we talked about. This muscle here, you see striations there. These other muscles are at a different focal angle. So when you section the muscle, this is essentially what it looks like head on, is where that's what it looks like when you're looking at it at a glance, essentially from the side. Very good. Back to our cartoon. So the thin filament, the actin, it is anchored in the Z disc, and there's a rod of nebulin that it swings around. It's, as we said, built to last. The myosin itself also has a protein that it essentially will assemble around. That particular protein is very, very large. In fact, the gene that encodes this protein is the largest gene in the human genome. It's called titin. Titin, it's huge, huge protein. It extends from one Z disc all the way to the other. And it has these regions at both ends that's highly unstructured. Because it's highly unstructured, it has the ability essentially to stretch and contract and to stretch and to contract and to stretch, contract, and stretch, and contract, and so on and so forth. So nebulin is the scaffolding for the actin thin filaments. Titin is the scaffolding for the myosin thick filaments. And so your orientation essentially of your myosin thick filaments and your myosin thin filaments is the same in both halves. They just are reflections of each other. That's the piece essentially that's loud. Essentially, that's the take home essentially from this picture here. So when the muscle contracts, the myosin, and myosin is a protein that has two parts to it. So there's a part that's globular, and that's the part that kind of looks like the head of a snake. Then there's another part to it that essentially is very fibrous. And those fibers essentially spin around each other. And that's why myosin is a dimer, at least myosin 2. Myosin 2 is a myosin you find in skeletal muscles. The myosin head has an actin binding domain on it. And it binds those actin microfilaments with that actin binding domain, and essentially binds it and it pulls it along it. It drags the actin along itself like that. So that's going to cause the sarcomere to shrink, to contract. The two Z bands will get closer together, just like what you see in this picture here. So we have a relaxed muscle in the top panel. Here we have a contracting muscle in the lower panel. Here we have a really contracted muscle in the bottom panel. Now take a look at that H band. Take a look at the top and the bottom. Tell me what happens to the H band. Yeah, it goes away. It completely goes away. Take a look at the I band. What do you see in the I band? I band is kind of shrunk as well, isn't it? How about that A band? That A band change? No, no. So the H band will disappear. The I band will eventually disappear. The H band disappears first. But the A band stays the same. The A band stays the same. And if we put this in a cartoon, you can see the two actin microfilaments getting closer and closer to each other. That H band is disappearing. And then as the two Z disks move closer to each other, the I band eventually disappears. That's what happens from on a micro level to the muscle. So if we use our smart art here and we just go through this, the myofilaments themselves don't change length, even though the total muscle length changes. So here's the muscle at rest. Now as it starts to contract, the two actin ends move towards each other. As they move towards each other, the sarcomere will shorten as the Z disks are brought together. They get closer and closer. Pretty soon the H band is going to disappear. The A band does not narrow, but the H band is narrowed and the I band has narrowed. So let's take a look at the so-called sliding filament theory of contraction. Sliding filament theory of contraction. This is called a theory, but the evidence for it is really good. When we say theory, we don't mean, oh, I have a theory of who killed Kennedy. No, when you say I have a theory of who killed Kennedy, you're saying I have a guess. No, it's better than a hypothesis. Theories in science are better than hypotheses. Theories essentially are things that are very, very well established. So when you look at this and you see theory, don't say, oh, it's a guess. No, it's not a guess. It's not a guess. So if we start at one, this is a resting muscle. You can see that the myosin head essentially is above the thick filament. The myosin has the head. The myosin, by the way, is a dimer, but it's not formed as a dimer here. We're just learning about one of them. There's another one on the other side of it doing the same thing. The C terminus, the carboxy terminus of the myosin, you see, forms that snake-like structure, that caduceus-like structure. That's called a coiled domain, for those of you who are interested. And it's just above the filament, but it's not caught, it's just kind of resting like that. So the first thing it's going to do is it's going to lose the phosphate. There's an ADP and a phosphate that's bound to it. It's going to lose the phosphate. When it loses the phosphate, you get the cocking and it binds actin. So the actin binding domain now will bind to the actin. Next, you're going to release ADP. When you release ADP, now the actin, which is bound, the myosin is bound to the actin, is going to undergo the power stroke. The head is now going to change conformation like this. So it goes from here to here to here. And it's going to drag the actin with it. When it cocks like that, that's the power stroke. Grabs the actin moves the actin moves this way. If it's going to do another power stroke, it needs to release the actin. When it binds ATP, it will release the actin. Then it will hydrolyze the ATP and go back to the relaxed conformation. It'll ditch the phosphate again, rebind, spit out the ADP, another power stroke, bind an ATP, reconc. Hydrolyze, spit out the phosphate, bind, spit out the ADP, power stroke. ATP binds, and then there you go. Now, how many of you have heard of a condition called rigor mortis? What's rigor mortis? After somebody dies, the muscles get really stiff. Reason being is that after you die, ATP production stops. There's no ATP to take the myosin off the actin. Hence, the muscle is very stiff. After a time, the muscle starts to break down because of bacterial activity, and the rigor mortis stops. So the rigor mortis will usually set in usually about 8 to 12 hours after somebody dies, and then it will last usually for a few days. And then that's why rigor mortis can be used to time how long someone's been dead sometimes. Now, this particular picture leaves out a complicating factor. And that complicating factor are these molecules here, troponin and tropomyosin. So tropomyosin, as you can see, is a fibrous snake-like protein, and it snakes across the actin. And attached to tropomyosin and attached to the actin is a globular protein called troponin. Notice troponin has 3 main subunits. So there is the troponin C subunit, there's a troponin T subunit, and a troponin I subunit. The troponin T subunit is going to bind to the tropomyosin. The troponin C subunit binds calcium, and the troponin I subunit is intermediate between the two. Hence the name I for intermediate. Well, what does this have to do with contraction? Tropomyosin covers the myosin binding site on the actin. So myosin, even with ATP, can't bind because the site where myosin is supposed to bind is hidden. When troponin binds calcium, troponin undergoes a remarkable conformational change. That conformational change causes it to drag the tropomyosin with it because those two are connected. Because the troponin T subunit, it's T because it's bound to tropomyosin. That conformational change drags the tropomyosin off the myosin binding sites of the actin. And now myosin can bind to the actin. And as you see here, again, picture shamelessly stolen from your book. Troponin binds calcium, it changes conformation, and now myosin can do its thing. Its thing is binding the actin and moving it as it undergoes that conformational change driven by the hydrolysis and rebinding of ATP. Myosin is one of these group of proteins we call a molecular motor or a motor protein because hydrolysis of ATP causes it to change conformation and do work. So here's a resting muscle. Myosin binding sites are safely hidden by tropomyosin. Actin, as you can see, is in the resting conformation. The signal for the muscle to contract will cause a release of calcium. We'll show you how that happens here in a bit. Calcium binds troponin. Troponin changes conformation, drags the tropomyosin off the myosin binding sites of the actin. Actin is going to release the phosphate and form a cross bridge with the myosin. Then it releases the ADP and you get the power stroke. That moves the actin. Then myosin binds ATP, that causes it to release itself from the actin. Hydrolysis of ATP will move it back into the resting configuration, and it's ready to go again, essentially. If, however, there's not enough calcium in the cell, calcium unbinds from troponin, and then the tropomyosin will move back over the myosin binding sites of the actin, and contraction has ended. So calcium is going to play a really central role in this whole process, as is ATP. Now, where does that calcium come from? It comes from two sources. It comes from the inside of the cell and the outside of the cell. Now, we talked about the plasma membrane of the muscle cell having these little passageways, these little honeycombs, kind of secret passages, if you will. And those are transverse tubules, or so-called T-tubules. And calcium can come from the outside into these T-tubules and can be let into the cell very effectively. Most of the calcium for contraction, however, is going to come from the sarcoplasmic reticulum. Now, if you look at the sarcoplasmic reticulum in the muscle cell, wherever you have a T-tubule, you have these inflations of the sarcoplasmic reticulum. They're called terminal cisternae. Terminal cisternae. And they're connected by cytoplasmic connections to the other one. That is associated with the other T-tubule. Most of the calcium is stored inside that sarcoplasmic reticulum. And here's essentially how this works. So you have at the motor end plate a variety of nicotinic acetylcholine receptors. These are ligand-gated ion channels. When they bind acetylcholine, they open, and sodium comes in. That's going to cause a local depolarization of that little patch of membrane. But also in this patch of membrane are voltage-gated sodium channels. You depolarize those, you get them to the threshold, they open, and now sodium runs into the cell, causing a robust depolarization. Depolarization is going to spread throughout the membrane. And notice that you have these here as well, and then you have them over here too. So when you get that depolarization that occurs in the T-tubules, you also have in the T-tubules voltage-gated calcium channels. But those voltage-gated calcium channels are covalently, not covalently, but physically linked, physically associated with sarcoplasmic reticulum release channels. And when these open and let calcium in from the outside, these open too and flood the sarcoplasm with calcium. So whenever you get depolarization of that membrane, you're going to have calcium not only brought into the cell, the internal source of calcium also going to be released as well. And that release of the calcium is going to flood essentially these contractile elements. They will bind to the troponin, it will move the tropomyosin off the myosin binding sites of the actin, and ATP will be allowed to let myosin initiate contraction. And so as you can see here, you have the action potential. The action potential again is initiated by the release of acetylcholine from the motor neuron that binds to ligand-gated nicotinic acetylcholine receptors. Those will cause a depolarization of a patch of the membrane, so they cause a greater potential in the patch of the membrane. If nearby voltage-gated sodium channels get those to threshold, they will open. Sodium comes into the cell, you depolarize the membrane, that spreads pretty quickly down the membrane, eventually causing the opening of voltage-gated calcium channels that are physically linked to release channels in the sarcoplasmic reticulum that line the terminal cisternae. Calcium now floods from the outside and the inside into the sarcoplasm. And we can see calcium rushing into the sarcoplasm here as it rushes into the sarcoplasm. We now get the mechanics of contraction occurring. Just like you see here, which we've gone over already. All right. Da-da-dee, 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 da. Oh, your book uses the term G-actin. G-actin. The use of that is a little bit unfortunate. Actin can exist as a monomer, in which case it's globular actin or G-actin. When actin forms a microfilament, then it's filamentous actin or F-actin. So by calling it G-actin, I think what the book is trying to tell you is that the binding sites on that actin are the same as it would be if it was globular actin. But this actin is in a filament, so it's not G-actin, it's F-actin. So also some students confuse G proteins with G-actin. They're not the same. They're not the same. So, all right, that's it for today.
I enjoyed it. It was wonderful.
I enjoyed it.
I like watching Dane suffer.
Yeah. Oh, I know.